digestive physiology. Don't forget, you have to drink in the lab. It's got to be elsewhere. Yeah, thank you. Then we see the video. So the models are out if you would like to review them. The torso models for digestive are out here. The reproductive models are there. There are obviously open lab times throughout the week, and there's actually more than usual this week. There's even weekend times. If you look on the doors up and down the hallway, you'll see the extended finals week hours for open lab if you want to come and look at a model. So, as I said in lecture, I apologize. I'm going to use this instead of the board today because my shoulder's screwed up, and I've been ordered by my acupuncturist not to do that for a few days, and so I'm doing this. So hopefully this is visible. We just have a couple last hormones to cover with respect to female reproduction, specifically with regard to lactation. And we're going to look at two hormones there. Don't eat those. We're going to look at two hormones. One of them is going to support this, and one of them is going to support this. So when you look at lactation, you're really looking at two phases: milk secretion, which is the actual manufacture of milk, and then milk ejection, which is the delivery of the milk to the infant. And there are hormones responsible for each. So prior to lactation, or during pregnancy, the high levels of estrogen and progesterone are going to get the tissue of the mammary glands to sort of kind of wake up. These cells were dormant. They were in G0 of cell cycle prior to this. You hit them with enough of these sex steroid hormones, and they're going to kind of go back into cell cycles. There's going to be more of them. They're going to start actively secreting or being capable of secreting their product, which is milk. Now remember, milk is a combination. It's kind of a complicated mixture of materials. It's mostly water. There's some ions in there. There's fatty acids. There's cholesterol. There's proteins, including antibodies. There's sugars like lactose. So it's basically customized nutrition for the infant. If you might recall this from last semester, mammary glands are actually just, they're skin-associated glands. They're modified sweat glands. So milk is actually just modified sweat, the strictest way of speaking. So I always invite people to go to Stewart's for a sweat shake after class. Kind of what it is. But yeah. So the hormone that triggers this is one called prolactin. It promotes lactation. Prolactin. It comes from the anterior pituitary. And it's being produced late in pregnancy and then after birth, and it will continue to be produced as long as necessary. So if you breastfeed, the neurological stimulus of breastfeeding tells the pituitary to keep on producing prolactin. If you stop breastfeeding, then that will stop. So it's supply and demand. You know, there's a positive feedback loop there. So theoretically, you can breastfeed indefinitely as long as there's enough prolactin stimulus. So anyway, that's what's causing this to happen. The ejection of the milk, the delivery of the milk, is going to involve some smooth muscle contraction around the ducts of the mammary glands. Also the suckling of the infant, but you also have to help. You have to sort of eject the milk, and that's going to be the job of oxytocin, which is this very same oxytocin that you saw causing the myometrial muscle to contract. It's good at making smooth muscle contract, and no matter where it is, it works. So you've already been making oxytocin, and again, you'll continue to make more of it as needed. As long as you breastfeed, you'll keep making oxytocin responsible for this. So I just needed to name those, or we already did name one of these, but I needed to get those two hormones out of the way. You already know this, but I'll tell you again anyway. Oxytocin is manufactured by the hypothalamus, but it's stored and released by the posterior pituitary. It's one of only two hormones that comes from the posterior pituitary. Oxytocin is one. The other one is ADH, right? Good. You're getting ready for the final, you know. So, excellent. So now, got those hormones out of the way, now we're good to go. Okay, questions? Awesome. So what I did with the class yesterday, the lab yesterday, was we good enough, right? What we did is we covered some digestive physiology, and what that's going to do is it'll free up next Wednesday. So you have the exam on blood and repro on Monday. We have one more class meeting in lecture, it's Wednesday, but instead of trying to shoehorn some digestive physiology in at the last minute, we can do some today, still have lots of time for the movie and the models, and that leaves all of Wednesday next week just for questions. So we won't have to cover anything new next week. So this won't take that long, believe it or not, because you already know the gross anatomy. We did it last week: esophagus, cardiac sphincter, stomach, pyloric sphincter, all that stuff. So I'll remind you of it as we go along, but I know you've seen it already. What I want to do is tell you about the physiology that's going on in those chambers. So I will draw some sort of a vague diagram. Here's your esophagus. Here's your, what's your esophagus? Here's your stomach. There's your cardiac sphincter and your pyloric sphincter. Close enough. Here's your small intestine. It's way longer than what I'm drawing, but pretend that's your small intestine. There's your ileocecal valve. And then here's your large intestine, which also is larger and not shaped this way, but just for schematics, there's your large intestine and there's your digestive tract. A couple other features that we mentioned last week in lab, anatomically. You got a liver sitting here. You have a gallbladder attached to that liver, and somewhere over here you have a pancreas. Label these guys. So here's your small intestine. Here's your large intestine. So y'all know those parts already. Let's talk about physiology, and then I have to name 4 hormones that control digestion, and that's it. So, physiology, we can talk, when we talk about the digestive system, obviously, you want to digest the food, and then you want to absorb the nutrients that you get when you digest the food. You got to account for all of that. The digestion may be described as being either mechanical, physical, if you want to call it that, or it can be described as being chemical. Your physical digestion, mechanical digestion, means you just take big food and make it into smaller food. Chewing food, for example, would be an example of that. Chemical digestion means you're actually doing chemical reactions. You're breaking chemical bonds. You're going to take protein and break it into amino acids. You're going to take a complex carbohydrate and break it into monosaccharides. You're going to take a triglyceride and break it into its components, etc. So all of that needs to happen. So let's start and see what happens in which chamber. Somewhere up here is your mouth, and obviously your mouth has teeth, and mechanical digestion happens when you chew food. I don't think there's anything too complicated about that. But there's also a little bit of chemical digestion that happens even at the level of the mouth before you swallow the food. In your salivary glands produce saliva, which is mostly water, but there's some enzymes in there. So your salivary glands, you got a pair of them under here, you got a pair of them over here, and there's a pair that I can't quite point to because they're back there. But you have these paired salivary glands, and among other things, the salivary glands are going to produce a chemical called amylase. Amylase digests starch. Starch is a carbohydrate. You knew that already, but starch is a complex carbohydrate. It's a polysaccharide. It's a polymer of glucose molecules. And the bonds that hold the glucose molecules together are going to be covalent bonds, and there are specifically, that was my next question, good, glycosidic linkages, right? So you get glycosidic linkages, you need to break those, because you can't digest a big starch molecule, you can't absorb that, you can absorb individual glucoses, so break those bonds. Amylase starts that happening. You're going to get some help later on, but amylase allows you to start breaking those linkages in the starch molecule. If you took a really starchy food, like a potato chip or like a cracker, plain cracker, and chew it for a really long time, it'll start to taste sugary because you're breaking starch into sugar. Anyhow, there's another enzyme that's relevant in saliva, but it's not there for digestion proper. It's called lysozyme. Lyse means to cut something apart, to break something apart, to split something, whatever. So lysozyme is an enzyme that breaks apart bacterial cell walls. So this is a disinfectant. If you happen to eat food that is contaminated with a potentially hazardous bacterium, this is a way to help prevent a future problem from happening. It doesn't sterilize the food or anything, and certainly many of the bacteria on your food are not harmful to you, but it's a way to help you avoid things like food poisoning. You got some lysozyme. The reason why some animals lick their wounds, there's actually several reasons, but one has to do with lysozyme being in their saliva and acting as a disinfectant. But anyway, in terms of pure digestion, you start on the carbohydrates. You really don't digest proteins, you really don't digest lipids at the mouth, and you certainly don't really absorb any nutrients at the mouth, but we kick things off. Next chamber. The esophagus is really just a muscular tube that's going to push the food forward through involuntary contractions. It's actually under the control of the vagus nerve. Remember the vagus nerve? It's cranial nerve number 10. In other words, it's not just gravity pulling food into your stomach. You can be upside down and swallow food and it still gets into your stomach because you got muscular contractions here. You got the cardiac sphincter there that prevents the food from going backwards. The only time the food should go backwards is if you have a regurgitation reflex and that opens up. That's an idea that, hey, if I eat something toxic, I can get rid of it before it causes me harm. Not all animals can throw up, by the way. There are animals that are capable of vomiting. A horse, for example, cannot throw up. This is why they call it. A rat can't throw up. Rat poison, when a rat eats rat poison, the reason it dies is because it can't throw up the rat poison. If it could, it would have thrown it up and it would have been harmed. But so some animals can't throw up. We can. That's a useful thing sometimes. But under normal circumstances, this stays closed. You get the food into the stomach. The stomach, you start to do a few things to proteins. So here's what's going on at the level of the stomach. There are cells in the wall of the stomach that are going to produce enzymes. They are protein digesting enzymes. They include things like trypsin, chymotrypsin, pepsin. These are just various enzymes, all produced by and used in the stomach, that break peptide bonds. Not just any old peptide bonds, but certain ones between certain amino acids. So what you're going to do with these enzymes is take long proteins and chop them up into short chains of amino acids. Not necessarily individual amino acids quite yet, but you'll hack them into smaller, manageable pieces. So you've begun protein digestion at the stomach. But you need other help here. The enzymes that work at the stomach don't work well at the pH of your normal food, which is neutral-ish. These work well at a pH of like 1.5. So you've got to somehow change the pH of the stomach contents. So there are other cells in the wall of the stomach that simply throw protons, hydrogen ions, into the lumen of the stomach. So here's your stomach, and there are certain cells here, let's draw one of them in exaggeratedly large, that have little active transport mechanisms in the apical side of their membrane, and they will pump, in other words, actively transport hydrogen ions into the stomach. That's why it's so acidic in there. That's an active transport mechanism. It's called a proton pump. Remember, that's a proton. It's called a proton pump. So if you have something like acid reflux or an ulcer that's being aggravated by lots of acid, you can inhibit those pumps with a proton pump inhibitor, like Prilosec or Nexium or some of these other drugs that are some over the counter and some prescription. That's going to just slow this down. It's going to make the contents of your stomach less acidic if you need that to happen. But whatever, when it happens normally, that's the way it works. So you give these enzymes the right pH. A lot of times people say, oh, you need the low pH because that helps break down the food. No, it doesn't. It gives the enzymes the right pH so that they can break down the food. It's regulation of the environment. Okay. What else is going on in here? you really don't do much in the way of carbohydrate digestion or lipid digestion. You just start kicking off protein digestion here. There's one other catch here at the stomach. If you have such a caustic, acidic environment there, who's to say that's not going to just cause damage to the wall of the stomach itself? It would. So you need to form a protective barrier, a lining around the stomach wall so that the acid of the stomach doesn't ulcerate itself. So what's a good material to line that with to be protective? Mucus. So there are still other cells in the wall of the stomach whose job it is to secrete mucus. They're actually not called goblet cells, but they're analogous to the goblet cells of the trachea. They're doing the same thing. They're just called mucus cells, but they do the same thing. They're little mucus secreting cells. So you're all set. You got a protective lining, you throw in the acid, you throw in the enzymes, you start breaking up some proteins. You really don't absorb much in the way of nutrients at the stomach. You can absorb some small molecules that don't need to be pre-digested. For example, if you take aspirin, aspirin's a tiny little organic molecule. That's going to get absorbed right at the level of your stomach. You don't have to break it down or anything. It's small enough to get right through the stomach line. Alcohol. You drink alcohol, it's getting absorbed right away through the stomach wall. It doesn't have to be processed and sent to the intestine for absorption. But sometimes if you design a drug, you design it intentionally. You want it to absorb rapidly. You design it so that it's going to get through the stomach wall quickly. But nutrients, you're not doing a lot of amino acid absorption here, let's say. You're going to do that in the intestine. So let's move on to the intestine. Good with the stomach? So you got all this semi-digested food coming out of the stomach through the pyloric sphincter into the small intestine. That material, which is your partially digested food and lots of water, because water, the enzymes were dissolved in water, etc. So it's this kind of soupy material. It's called chyme. So that chyme, that partially digested stomach contents, is going to now be passed on to the small intestine. To be digested at the small intestine, of course, you're going to need enzymes, but the enzymes that work well there work well at like a pH of 8. And you're coming out of a thing that had a pH of 2, 1.5, something like that. You got to buffer that pH. You got to raise that pH. So, of all the things you can think of that we've covered already this semester, or that you might know about in general, name one chemical, one substance that would be really good at buffering an acid and raising pH. We all know it.
Might be right.
We've all seen it before a lot of times. Bicarbonate ions. They're great at buffer. Remember this reaction? I told you you'd see that 1,000 billion times. Yeah, you've seen this one before. I don't have to. You can see that in your sleep. This is a great buffer. If you have lots of these and you got too many of those, these will attach to those and become something else. They will remove bicarbonate ions. It will remove free protons from solution. So that is a bicarbonate ion. It is a very common buffer in biology. It is found in the stomach, it's found in the blood, it's found in oceans and lakes and soils, it's found all over the place in biology. It's a great buffer. It's found in your lungs, you name it. So you're going to put some bicarbonate ions into the small intestine so that you can buffer all that acid that is heading its way. So how do you get it there and where does it come from? It comes from the pancreas. There are specialized cells of the pancreas whose job it is to actively secrete bicarbonate ions into a duct that gets to the duodenum of the small intestine. So here's your bicarbonate ions being sent from the pancreas to the small intestine. Now you can buffer. Now you can raise the pH. Everything's good. What about the enzymes? Unlike the stomach, the small intestine does not make its own enzymes. It's got to rely on another organ, the pancreas. So there are other cells of the pancreas whose job it is to make various enzymes. and send them, I'll put ENC for enzyme, and put them into the small intestine. Those enzymes include lots of different types. There are lipases to digest lipids. There are enzymes that'll break down carbohydrates. There are enzymes that'll finish off the protein digestion. There's a whole cocktail of enzymes there that'll, take care of everything you need. And again, it's specialized cells of the pancreas doing that. Different ones than made the bicarbonates. These are all those, remember the pancreatic acini? Those cells, they're the cells that are doing this stuff. Okay. Enzymes, ENC. Yeah, it's supposed to be. Just as a review, I know we're talking about protein and carbohydrates so far today. Triglycerides are common foods that you eat in your diet. They're complex lipids. If you break down a triglyceride, what parts do you get? Glycerol and 3 fatty acids, yes. Yesterday's class didn't get that, and I played hangman with them, and they still didn't get that one. So yes, you get fatty acids and glycerol. Remember, a triglyceride is made of 1 glycerol molecule and 3 fatty acids. That was from back a while ago, so I'm sitting with the preserved things. Formaldehyde all over and over. Anyway, yeah, go right back. So you do some pretty significant enzymatic digestion at the level of the intestine, thanks to the pancreas. Because again, the intestine can't make its own enzymes. Just give me 1 second. Thanks. I'll bring it back in. There's one other catch though. So you got the right pH, you got the enzymes. They're going to take care of all the rest of the digestion you need to do. But think about the lipids. This is just a lot of water with all these things dissolved in it. If you have a lipid, cholesterol, triglyceride, fatty acid, whatever, and you want to break it down, the enzymes aren't going to be able to get to it. The enzymes are dissolved in water. They're hydrophilic. This is a glob of hydrophobic stuff. You're not going to be able to get them to talk to each other. So what do you have to do to the lipids to get them to mix with the water so that they can be subject to enzymatic breakdown? Emulsify them. That's exactly it. You need an emulsifier. So, the emulsifier that's going to get dumped into the small intestine is bile. Remember bile? You had me for bile 105. That was one of those extra credits. Bile.
I'm sorry, could you go over the term emulsify?
Emulsify. To emulsify a fat is to, because fats are normally hydrophobic, right? So to emulsify a fat or lipid means to make it miscible in water, to allow it to mix with water. It's technically not dissolving in, but it's to allow it to mix well with water. So for example, soaps act as emulsifiers. They take the grease on your hand or on a greasy plate or whatever and allow the water to mix with it so you can get rid of it. There are emulsifiers in foods. If you want to get oil and vinegar to mix in salad dressing and not separate, you put an emulsifier in and it'll blend together. Here, I want to emulsify the fats that I just ate so that the enzymes can rip them apart. And the emulsifier that you employ is bile. Bile is made by the liver, and it's stored by and then ultimately released by the gallbladder. It goes through a bile duct and gets to the duodenum, the beginning of the small intestine. Now I got everything I need. I got all the enzymes to take care of all the food I've eaten. I've got the right pH for them to work, and I've got an emulsifier so that the lipids can be subject to enzymatic breakdown. I'm in business. I just need some space now. And you're 27 feet long if you're a small intestine. You got plenty of space. Okay. So you do the digestion, and the results, the sugars, the fatty acids, the amino acids, all the breakdown products, will be easily absorbed through the wall of the small intestine because A, it's 27 feet long, there's a lot of surface area there, and B, rather than just having a smooth wall like that, the wall of the small intestine is wrinkly like this, which gives you even more surface area. The little wrinkles, you're probably familiar with the term, the wrinkles are villi. And that wrinkliness, as opposed to just smoothness, like would be the alternative, gives you plenty of surface area. You're going to absorb everything you want to absorb. Amino acids, sugars, fatty acids, ions, you name it, whatever is useful stuff. So that's your small intestine. Now, the other thing to note about the small intestine, it's got plenty of bacteria in it, and they're supposed to be there. The stomach should be bacteria-free. But the small intestine is supposed to have a flora of bacteria there. Your large intestine also should have a flora of bacteria. Some of them are helping you digest. Some of them produce enzymes that you can't produce, and they're helping you break down food that you would otherwise not have been able to break down. Some of them are doing completely different things. Some of them, for example, make like vitamin K. Vitamin K is produced by bacteria in your gut. You don't make it yourself, but they're there to help you. So there's a lot of reasons why there are helpful bacteria in your gut. E. coli, Enterococcus, things like that. So what leaves the small intestine is going to be a bunch of stuff that you couldn't digest, fiber, things like that, cellulose. A bunch of stuff that you don't want to absorb, excess ions that are more than what you need, let's say, waste materials, leftover bile, which may have some garbage in it that you're done with, you don't want, bacteria, and a whole bunch of water. So the large intestine, the colon if you prefer, has a couple of jobs, but tops on the list is get back some of that water. If you just take what comes out of the small intestine, run it through the large intestine without doing anything to it, you're going to have dehydrating diarrhea and you're going to die from it. The number one killer in the world is diarrhea. It's dehydration from diarrhea. People, most people in the world don't have access to clean water. And if you have diarrhea, you lose water this way, you dehydrate, you're dead. Especially if you don't have other medical care. So you better have a large intestine when it's working in a healthy way that can reclaim a lot of that water. Because remember, you took a lot of water, the enzymes were dissolved in water, the bicarbonates were dissolved in water, etc. You put it in here, you got to get it back now. It did its job, you don't want to waste it. So a main function of the large intestine is to just take back some water, put it back into your blood so you don't dehydrate. Now, along with the water, sure, you bring in some ions back. For example, potassium ions are brought back here, etc. But your main job in the large intestine is reclaim that water. By the time you get to the end of the large intestine, what's left is all the indigestible stuff, the fiber, etc., a little bit of water, and lots of bacteria. But that's okay because bacteria reproduce really fast, and under normal circumstances, you're not going to run out. They'll just keep making more. Some people take the probiotics to sort of seed even more bacteria into your gut, and that's good, that's fine, that's healthy. But what becomes feces is basically the dehydrated leftovers of what came out of the small intestine, minus a couple minor components. And so that's what happens. So we brought all of these into the picture, all these into play. Any questions? All right. And all I have left to do is I'm going to walk you through that and tell you 4 hormones that significantly affect all the steps we just looked at. There's more than 4 that play a role in here, but there are 4 primary, what I like to call gut hormones. By that I mean not only do they affect the digestive system, they're also made by the digestive system. So yes, digestive organs are capable of making some hormones. So let's go ahead and start.